Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore different type of respiratory substrates. At the end of this lesson, you will be able to explain the relative energy value of carbohydrates, lipids, and protein as respiratory substrates. Let's get started. While glucose is the primary respiratory substrate for certain cells, such as neurons in the brain, red blood cells, as well as a lymphocyte, other cells can also oxidize lipid as well as amino acid for the energy. Triglycerides, for example, are broken down into glycerol and fatty acid. So the glycerol is converted into a triphosphate, which is an intermediate in glycolysis. And glycerol, as we know that it is a 3-carbon compound. The majority of the energy in triglyceride actually is stored in the fatty acid. Beta oxidation occurs and break down the fatty acid chain into 2 carbon fragments. And these 2 carbon fragments combine with coenzyme A to form acetyl-CoA. And coenzyme A carry this acetyl group into the Krebs cycle. How about amino acid? So amino acid in excess are converted by enzyme into intermediates of glycolysis as well as Krebs cycle. But before they can enter into the glycolysis or the Krebs cycle, the amino acid must undergo a process known as deamination, where the amino groups of the amino acid is removed. So this diagram summarizes the process of how these different type of respiratory substrate enter into the aerobic respiration pathway. So you can see that the protein is hydrolyzed to form amino acid. Deamination takes place where the amino group is removed to form ammonia, and the remaining keto acid will act as the intermediate compound in the glycolysis as well as the Krebs cycle. Regarding to the fat or triglyceride, the hydrolysis of triglycerides form glycerol and fatty acid. Glycerol is converted into the triphosphate, which is indicated by glyceroldehyde 3-phosphate here, and continue the glycolytic pathway. The fatty acid undergo beta oxidation to turn into a 2-carbon fragment, the acetyl group, combined with the coenzyme A and enter into the Krebs cycle. Let us look at the energy value of each type of respiratory substrate. The majority of energy released during aerobic respiration comes from the oxidation of hydrogen atom to form water. So reduced NAD and reduced FAD transfer their electrons to the electron transport chain. So before they can do so, reduced NAD and reduced FAD must release hydrogen atom, and this hydrogen atom splits into protons and the electrons. Therefore, the more hydrogen atom present in the structure of the substrate molecule per mass, the greater the energy density. So energy density refers to the amount of energy available per unit mass of a respiratory substrate. So the energy density of a respiratory substrate is determined by burning a known mass of a substance in oxygen within a calorimeter. Be careful in this case, the spelling of the calorimeter, don't confuse it with the calorimeter where we learned this in our AS topic. So in this calorimeter, so inside the chamber there, we can actually burn the known mass of, for example, carbohydrates, lipids, as well as a protein. And due to the release of the heat energy, this heat energy transfer to the water. And by using this thermometer, we will be able to measure the increase of the temperature. Okay? So by using a particular formula, we will be able to calculate the energy release. So we know that lipids have the higher energy density if you compare it to the carbohydrates or protein. So why fatty acid have more energy or higher energy density? It's because fatty acid have relatively more carbon-hydrogen bond than carbohydrates. So therefore, we do know that there is more hydrogen atom per unit mass in fatty acid compared to the carbohydrate as well as protein. During the oxidation of fatty acid, more reduced energy molecules are produced compared to glucose metabolism. So there are more hydrogen atoms available to build up a proton gradient across the inner mitochondrial membrane. So this increases the stiffness of the proton gradient, which enhances the chemiosmosis and oxidative phosphorylation, which results in higher productions of ATP during aerobic respiration, particularly oxidative phosphorylation. So this table summarizes the energy density and some remarks about this respiratory substrate. If you look at the carbohydrate, generally energy density is about 15.8 kilojoule per gram of carbohydrate. And carbohydrates are preferred for quick energy release, particularly in the cells like neurons and also red blood cell. How about lipid? You can see that the energy density of lipid is 39.4, more than the double of the carbohydrate, right? So lipids have the highest energy density among carbohydrate, lipids, and protein. So which is why they are used for long-term energy reserve in organism. And don't forget, lipids can actually serve as energy reserve, particularly the oxidations of lipids produces metabolic water, which is a water source for those animals live in the environment which is lack of water. How about protein? So roughly about 17.0 kilojoule per gram, okay, of energy released by 1 gram of protein. So similar to lipids, protein actually serve as the long-term energy reserve as well, but protein are usually used for growth, repair, and enzyme function. But yeah, when we need the energy, it can be metabolized for energy when necessary. So with that, we have come to the end of this lesson. I hope you have found this lesson helpful. If you have any question, feel free to leave them in the comments section. See you in the next lesson. Have a great day.